Me, I was a former graduate student at Stony Brook, and I studied for my entire dissertation diadromous fish in the Carmen's River. So this is a subject close to my heart. And since then, I'm teaching over at Suffolk County Community College, and we've been able to get some Suffolk students involved with the Peconic River runs, involved with Elizabeth at Peconic Estuary Partnership, and we've been able to actually estimate the run size over at the Peconic River. One of the students was phenomenal and really just took over this project and was able to present this research at the National AFS meeting this past September virtually. So this presentation is a modified and updated version of that. So at any time, if anything's unclear, just let me know. So I won't go into a lot of background. I'm sure everyone's very accustomed and knowledgeable about alewife, but I do want to point out that in Long Island, our estimates of run size are really lacking. There's a big data gap. And if you look at the run sizes across the East Coast, they're fairly well established and fairly well studied, but for some reason, Long Island just kind of gets skipped over. I think it's because we have smaller rivers, but we have a lot of them and they do add up. So alewife here in Long Island generally arrive in waves beginning in February, sometimes into March, and usually stick around till June. And they do have this really complex migration behavior. A lot of my research and other researchers have established that they spawn multiple times throughout their season, which really complicates counting them. Traditionally, we thought they were a single spawner where they move upstream and spawn and leave. And now we're realizing that they can do these oscillations and move back and forth often on the spawning grounds, which again, makes counting them very difficult and estimating their population size difficult to be accurate. So when we're counting them and estimating their population, we're usually going to count them at sites of fish passage. It's convenient for everyone, and we can do it through either tagging studies or camera surveys. And if you're not familiar with the Peconic run, right over here is the on the lower bottom is the Grangerville Fishway at Riverhead. So here's the Peconic River. So generally, and correct me if I'm wrong, but from what Byron has said and from what the literature has said, it's a primarily alewife dominated run. There's not too much evidence of blueback herring, so I'm just going to call it an alewife run. Even though we know blueback herring does occur on Long Island, it's not too common. But there was a Nature Lake Fishway built in 2010. This restored about 25 acres of spawning habitat, which is fantastic. But we are looking at future fish passage projects, and they are underway, and hopefully we'll get them up and going. But before we do that, we really need to understand what the population is doing, and we need to establish a baseline before we continue restoring these fish passage to understand their efficacy and to make sure they're actually working. So here's the Peconic site. Here are two students, Anna and Jenna, maintaining the camera. It's a fairly small structure, and we have the camera maintained about 2 to 3 times a week. It was installed in both 2019 and 2020. In 2019, a student from the MCPU program over at SOMAS analyzed the run. And in 2020, we helped analyze the run. So the students were really gung-ho about it, and they were getting out in the field early. And then, of course, COVID struck and the world shut down. But we were able to continue monitoring it. Elizabeth had to go out there every week, but the students were able to analyze the data, and we were putting it together. So the camera was recording 24/7. And just to give you an idea of what it looks like, here's a little clip of a video of fish going through. So this is fairly clear, and it's fairly good footage, and you can see a bunch of fish going through. So we were recording every single fish that went through in 10-minute time segments. So overall, we had 3 main objectives. One, estimate the annual run size. That was our main goal. But we also wanted to see if there were any temporal trends or maybe environmental trends that could drive the run and maybe help us predict when we were going to see peaks in alewife. We also wanted to evaluate our subsampling method because watching the entire run is very exhaustive, it's labor intensive, and it's difficult. So we had to use a subsampling method, and we wanted to really see how well we did. So for the run, we subsampled videos from each day. We broke the day down into 4 separate periods. Subsampling is not necessarily a straightforward thing. There's no standard subsampling method that I know of. Some people just do random videos throughout the day. Some people break it into periods. We know there's a temporal trend here, so we broke it down into 4 periods, 6 hours each, starting at 12 a.m. to 6 a.m., 6 a.m. to 12 p.m., and so on and so forth. From there, we selected 5 segments from each period, and then overall, we were watching 14% of the run per day. And then just to get the total run estimate, we were just extrapolating that out to try to get a rough estimate for the entire run. So within that video analysis, in a 10-minute segment, we wanted to record the number of fish, the species. We obviously couldn't tell if it was a blueback herring or an alewife, so we just assumed alewife. But we did catch some other fish on there. And we also wanted to record the direction. So here is our little box representing the camera. So ideally, you would see a little fish go through, and that would be our upstream movement. However, there were other movements documented. Fish go downstream. Sometimes we have fallback where they enter the field of view and then return back downstream. Sometimes they do the opposite and go fall up, where they enter the field of view from upstream and then return upstream. So we wanted to record all of that, but for our run size estimation, we were only using upstream counts. And to give you a sense of some of the chaos that we saw in here, this was from the first day of recording, and there's like 50 fish going back and forth through this one little clip. So that's why I thought it was important to record the direction. So overall, it was successful. We analyzed the whole run and estimated around 58,000 alewife going through, which was a great news because in the 2020 run or the 2019 run, we had a 67% increase. So that was, really encouraging. Our largest peak on 3/19, fairly early, we estimated around 4,200 fish going through. And this is crazy to me because when I analyzed the Carmen's run, the total run size going through the fishway was around 2,000. So we doubled that entire population in one day of the Peconic River. And so in this graph, hopefully you can see the different colors on the bars, and these represent the different periods. So not every single period is reflected evenly. The proportions of periods are inconsistent, so there's going to be significant temporal trends in our run. Another way of graphing this is a cumulative distribution. So we want to see how the run progresses throughout the season. So on the bottom is 0, and as you go up the vertical axis, it gets to 100%. So you can see how the percent accumulates throughout the run. And what we saw was we recorded the run from 228 to 518. We could have missed some early fish, but if you put 95% confidence intervals on it, the majority of the run was completed between March 8th and May 4th. So I think we captured the run fairly well. We also can see from here that the halfway point occurred really early, March 26th, which was kind of unexpected. So you can see the bulk of the fish were going through in the first few weeks of the run, whereas after this point, they kind of petered in and probably went in smaller waves. You can compare this to temperature, and we did see some correlation to temperature. The run began around 5 or 6 degrees Celsius, which is pretty cold. We were expecting a little bit higher. And then the halfway point occurred at 7 degrees Celsius. So it's occurring at a little bit cooler waters than literature has said. The run ceases around 15 degrees Celsius. We also looked at some lunar data. We couldn't find any correlation between peaks of alewife and the new moon or the full moon. And then we also would eventually like to pair this up with some tidal data, but as of right now, since it's subsampling data, we don't have that fine scale to match it up with tidal wave or tidal points. So that's in the future for now. So overall, we did experience really strong temporal trends. And what we found was 50% of the fish went over in the dark at night, which was really significant. Very few fish went over during the day. And we did see peaks at dusk and dawn. Now, the way I organized this, I gave 2 hours for each dawn and dusk component. Obviously, this is going to change as the season progresses. And again, as we get more fine scale data, I think we'll improve upon this. But this significantly impacted how the periodic proportions came out. So overall, 83% of all alewife traveled between 6 p.m. and 6 a.m., which is overwhelming. And that meant during the day between 6 a.m. and 6 p.m., not many fish were going through. So this is gonna, influence how we subsample and how we kind of model the overall population going forward. So we want to break down by amount of fish that we're going over by hour, because maybe we can improve our subsampling. So here in this graph, it's the mean number of fish from each 10-minute sample per hour. And what you should see is there's significant trends. So in period 1, it's around 6.5 fish, high standard deviation. Period 2 and period 3 during the 6 a.m. to 6 p.m. hours, fairly low. And then in period 4 during nighttime, pretty high. However, we have one problem child, and that's the 6 a.m. hour. This is an outlier. It's significantly different from every single hour in that period. So this probably means we need to potentially regroup the hours into different periods so we can more accurately subsample this data. So after we completed our subsampling survey, we wanted to evaluate how we did. So again, this takes a long time. So we chose 3 days. We obviously want to do more, but it does take a really long time. So we chose the highest count day, which was March 19th. And then we chose 2 other days randomly, one before and one after. So March 9th, which was very early in the run, and April 15th to do a complete 24-hour analysis. And we wanted to compare our observed to predicted. We also wanted to see how the hourly proportions were laid out. So how did we do? So first we have March 9th. Overall, not many fish were going through, roughly 450. We observed 450 or so. And our prediction was fairly close. We had the same kind of proportion per period. We did underestimate by 19%, but that's okay. I'd rather underestimate than overestimate. March 19th, little different story. This was our peak run. We slightly overestimated by 23%, but that's to be expected. There was a lot of fish going through. It's our peak day, but the proportions for each period look similar. So I think we're on a good track. And then this past week, we finally finished April 15th, which just threw us for a loop. And unfortunately, we overestimated by 120%. So this is not great, but why? Why did we overestimate? It looks as though our ratios are just not matching up between our observed and our predicted. And we really want to avoid overestimation, because this means we're thinking that the population is doing better than it is, and that's dangerous. So let's see what we can do. If we look at the hourly distribution for all of those days, here's March 9th. So I standardized the percent or the proportion of fish going through by each hour. And here we see a predominantly nighttime run with some fish going over in early morning. So this is March 9th. We add in March 19th. This is our busiest day. This is a more even distribution, so this is great. But again, we still have more fish going over at night and in the morning, not many going over in midday. And then we add in April 15th, which again kind of skews our estimates with just a lot of fish going through the 6 o'clock hour. And again, this is going to correlate to times around dawn. So this signifies a really important biological trend here. When we look at this compared to our average subsample data, it's really not picking up on some of these, these trends. So it's possible we're going to have to, redo our subsampling method to really accurately capture some of these either predominantly night runs or predominantly early morning runs. So overall, we saw that there's really high variability within periods and hours and days and proportion per days. So fish aren't just slowly coming in all at the same time. We are seeing waves of fish, and there is some sort of environmental trigger pushing them at certain times of the day. However, we do not see a lot of variability during the midday, which is good. So one of the ideas I've been playing around with is to split up this timing in between 3 different groups: early morning, midday, and evening. And when we do that, this significantly lowers some of the variance we see, and I think would optimally, give us the lowest sort of variance estimate on our population. So We need to do a little bit more analysis on this before we implement it, but this kind of shows us that pushing that 6 o'clock hour into the early morning group is advantageous. So overall, there's always going to be some pitfalls using camera data to estimate population size. And with error in the population estimate, some of it's going to come from our subsampling. Obviously, we'd love to watch the entire run, but sometimes it's just not possible. However, we do have the added complexity of multi-spawners. So what happens if you're counting the fish multiple times, the same fish over and over again? That means our population estimate's going to be much higher than it is. And again, that's dangerous. We want to avoid that. And when we're counting the same fish multiple times, we really need to understand what proportion of the population are multi-spawners. From the Carmen's River research, we saw that about 20% of all the fish were going over the fishway multiple times throughout their spawning season. In the Peconic, we have a little bit of a different story. The fishway has really, really high passage efficiency. So maybe we're going to see fish go over more than just twice, maybe 3 times, maybe 4. So overall, we need to understand what the proportion of multi-spawners are, and then how many times does a single fish spawn, or at least go over that fishway throughout a single season. So this is, especially important to establish before further fish passage is installed. I think that migration behavior could be a function of potential river distance or freshwater distance. If you have a 100-kilometer river, the likelihood of a fish going back and forth between, the highest spawning grounds and the brackish habitat is going to be low. But in a very small river that's only a few kilometers long, the ease of going back and forth between these brackish habitats and freshwater habitats is much easier. And then if you look at the Peconic fishway, passage efficiency is very high. Connectivity is very high. So the ease of moving back and forth in this area could be high and ultimately skewing our results to overestimate what the population is. However, if a fish passage facility goes in, that could increase spawning habitat and lower the ratio of multi-spawners, potentially. The whole thing is we need to establish what's going on. And thankfully, we're going to collaborate with Peter Daniel to look at his pit tag data and ideally get an idea of what the multi-spawner proportion is, how many times are they going over it, and kind of apply that coefficient to our overall population estimate to really fine tune it, because that's really, really important. And you can kind of see the antenna just upstream of the camera here. So that was working throughout the 2020 run, and I think the 2019 run, correct me if I'm wrong. So we can definitely fix the populations estimate. So finally, I wanted to end on just all the other really cool stuff that we were able to see. This camera provided us some extra video footage on glass eels going through. So we at least have some timestamps of when glass eels were in the river. We also saw some big yellow eels go down, and this was always really exciting to see. And if you check out that video, you can see something in the big yellow eel's mouth as it kind of rolls down the river. And then one of the favorites is watching a snapping turtle just fall down the fishway. That's always one of the best. I had to slow it down so you could see it. So sometimes it takes a while, but there it goes. And we were also able to see some other fish like killifish and gizzard shad, largemouth bass. So with that, I want to thank Byron. He was always out there helping and teaching the students how to, catch an alewife, Sarah from Peconic Estuary, Lauren from Cornell, all the students, Jenna, Anna, Brendan, Casper, Evan, they all dedicated a lot of time to this in COVID to analyze the video data. So we're continuing this this year, so I'm really looking forward to adding on to this research. So with that, I'll take any questions.